Hello, everybody. Here we go, starting off the semester with our first Anatomy and Physiology number 2 lecture. Couple of notes here. This is Dr. Jenkins. A couple notes before I jump right in. For those of you that had me for A&P 1, you've already heard me say this. If you had someone else, or maybe you haven't had A&P 1, hopefully you have, I just want to be very upfront, right? So in A&P 1, we started slow. We eased into the semester. We started off with an intro chapter with terminology, right? And then we did the chemistry and parts of the cell and the tissues before we even got into any body systems. And in A&P 1, some of the hardest material tended to be at the end of the course with the nervous system. Here in A&P 2, it's a different story. So for A&P 2, we're going to jump right in. So one thing that often happens is that it catches some students off guard. We're not used to being in class. We had some time off, and we kind of expect to be able to ease in. But I wanna caution you against that, because in A&P 2, some of the hardest material is actually at the beginning. Over the previous years, it tends to be that in A&P 2, the lowest exam grades I have are for exam 1, which is the cardiovascular system. So I know you can do it. I just don't want you to get lulled into, we're just starting easy. No, we're jumping right in. All right. So get ready to roll. Do what you know how to do. Study, work hard. Ask me if you have questions. Reach out. I can't help you unless you reach out, right? All right, so our very first chapter is the blood. So here's a little cartoon to start things off. Hopefully no one gets a D+ in the class, much less for their blood test results. Haha. Blood typing, that is. All right. So our first unit this semester is the cardiovascular or the circulatory system. We can really use those words interchangeably. And in this unit, we have 3 chapters. The first chapter, which we're going to start right now, is the blood. The next chapter we'll do is the heart. And then the final chapter we'll do as part of the cardiovascular system is a chapter on blood vessels. And that may seem easy. Well, blood vessels, arteries, and veins, but that one is sneaky hard because it includes blood pressure regulation. And we're going to get into the renin-angiotensin mechanism and a bunch of graphs and schematics. And I'm going to get really excited. You can totally do it. I'm here to lead you through the way, but I can only do part of it, right? I'm going to lead you through it the way that I think makes the most sense. I'm going to lead you through it. Hopefully this is still going to save. We shall see. I'm going to lead you through it in a way that is stepwise and progressive. So you should be all set. You just have to do your part, which is study, reach out if you have questions. So for the blood, this chapter is the blood only. Here's our outline. Of course, the introduction with the function. We'll talk about the actual parts of the blood, and then how blood cells are formed, and then blood clotting. Okay. Now, I just want to make a note on the overall functions of the cardiovascular system. So we know overall the cardiovascular system, it contains the heart, which is a pump, and really it contains a bunch of vessels that go out of it. And let's do blue to represent deoxygenated blood coming back in, and vessels that go to it. So really, the cardiovascular system is a mechanical pump, the heart, which pumps blood through a series of tubes called blood vessels. So in that way, it's very mechanical. What then are the functions? Well, the biggest function, one of them, is to transport. This is how we're going to pump blood out to the cells of the body. This is how we're going to pump blood to the lungs to pick up oxygen. But it's not only about oxygen when we talk about transport. The blood transports so much more, right? So much more than just gases. This is how we transport nutrients, right? Once nutrients are absorbed into our digestive tract, how are they going to get to all the cells of our body? Blood vessels. Hormones, antibodies, neurotransmitters, enzymes, I mean, you name it. If we need anything transported in the blood, it's going to occur through blood vessels. And that's also not including heat, right? We can help regulate our body temperature through transporting more or less blood to a certain area of the skin, right? So if we want to dissipate heat to get rid of heat, we can send more blood flow to the surface of the skin, and that moisture can evaporate. We also utilize the cardiovascular system for protection because one of the very important blood cells are white blood cells that help us with our immune system. We can also transport antibodies, and we can transport platelets, which help with blood clotting. And then there's one function that maybe you don't think of, but the cardiovascular system, because it's able to transport so much, has a key role in regulation. If we were in person, you would see me walking side to side in front of the classroom, probably up and down, maybe even through the aisles, because this is something that I get excited about. I'm going to be talking about it all semester. Really, when we talk about regulation, we're talking about homeostasis. You know what that is. I'm going to pause for dramatic effect. What? But I'm also pausing because I want you to remember what homeostasis is. You cannot forget, ladies and gentlemen, what we covered in A&P 1, because it all matters and builds on each other. We did this in chapter 1 of A&P 1, right? Homeostasis, the ability of the body to maintain balance. We have to be able to maintain balance of blood pressure, maintain balance of pH, maintain balance of blood volume, red blood cell count, and so forth. So every body system participates in maintaining homeostasis, but particularly the cardiovascular system, because it can transport so much, give some, take it away, transport this, transport that, plays a huge role, particularly in blood volume or fluid regulation and our ability to maintain pH and buffer against major pH changes. Make sure that you're reviewing the parts that I'm talking about. If you've had me before, you could pass this along to people who maybe haven't. When we come to exam time, the questions are going to come exactly from what we talked about, especially when I circle something or put a check mark beside it. If I write something down like this, you should review it. There may be instances, in fact, there will be instances, where I'll say, this is interesting, blah, 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 but you don't need to know it. And I will say you don't need to know it. And when I say that, I mean it. Okay? So I want to make use of your time and value it. So yes, people can learn by reading the book, but my goal here is to really make the learning process more efficient for you and better. So I want to make use of your time and make these really valuable for you and towards the exam. Otherwise, it would be disrespectful of me to ask you to listen to all these if they weren't going to be on the test. All right. Now, we've covered the overall functions of the cardiovascular system, but I want to talk specifically about the functions of the blood, because that's what this chapter is all about. So the blood, of course, is involved in transportation. Think of the blood as a fluid medium. If we didn't have the plasma in the blood, the fluid, we wouldn't be able to transport things through it. So that fluid medium, the fact that blood is a liquid, is very important. We talked about regulation, right? Homeostasis. So particularly, one thing I'm going to point out now is the blood's ability to regulate pH. Really important. We rely on the blood for blood clotting. The fancy word for that is hemostasis. We rely on the blood for defense or immunity, which we already talked about, folks. That's, of course, through the white blood cells. And then I didn't want to forget the idea that we can help to maintain body temperature. So that's one that we don't think of, but we would have a very difficult time maintaining our body temperature without it. So as I said before, our cardiovascular system is able to put more blood in certain areas. So if we want to get rid of heat, we can push more blood to the surface of the skin to help some of that water from the blood evaporate, which has a cooling effect. Okay, so review all of those. All right, here are some properties to talk about. As we're going to see, the blood has two parts, okay? The blood is made up of plasma, and that's really just the fluid, mostly water. And then the other part of the blood is formed elements, which really is comprised of cells and platelets. And we're going to get much more into this in a little bit. So our two main components of blood, plasma, which is mostly water, which is the fluid medium, and then our formed elements, which are our cells and platelets. Of that blood, the plasma and the formed elements, make sure you have an idea about what a normal blood volume is for a human adult. Usually for most people, we have at any given time somewhere between 4 and 6 liters of blood in us. If you think about 2-liter bottles of soda or seltzer, each of these is 2 liters. So maybe for an average-sized adult female, they have 4 liters of blood going through them at a time. Maybe for the average-sized adult male, he's got 6 liters going through him. It's pretty significant, right? All right, sorry about that. Just paused for a drink. You can read about the temperature. I'm not going to ask you about that. But of course, make sure you know the pH range of blood. I hope you already do, but if you don't, here's your chance, okay? Know it, know it, know it. The normal range, the healthy range of the blood, the pH, is between 7.35 and 7.45. So the middle, 7.4. This, unlike other fluids in our body, the blood pH is really strict. In other words, that's a really narrow, healthy range. And if we fall outside of that narrow range, boy, we have problems. Right? Or should I say, girl, we have problems. Or I should just say we have problems. Right? So that's a very narrow range for what's healthy. If we compare that to the urine, for example, which we're going to get to later, but I just like to compare. The urine, we can have a pH of urine anywhere between 5 and 8 and pretty much be okay with it. Because think about it. The urine is how we get rid of things. If we have too much of something, if we have too much of acid or too much of this or that, we can get rid of it through the urine. So we're okay with a wider range of pH in the urine. But with the blood, not so much. Later on this semester, we're going to have a whole section on pH regulation because it takes a lot to keep blood within that narrow range. And overall, we're pretty good at it. All right. Now, my friends, you're going to see in some of these slides in A&P2 little rectangles in the corner. Okay. You're going to see little rectangles in the corner. And those are little A&P1 reminders. These are not going to be on the test necessarily, unless I talk about it as part of the lecture. But just imagine that green, what I've put in a green rectangle. It's just me nudging you, right? Hey, don't forget, right? Don't forget we learned in A&P1. So it's my way of, in a friendly way, reminding you to don't forget it. Hopefully you can answer this question. If we were in class, I'd probably look at some poor little soul in the crowd, and especially on the first day, they'd be like, what? They'd be afraid. No need to be afraid. What type of tissue is blood? We talked about 4 main types of tissue: epithelial, connective, muscle, nervous. Who remembers what type of blood? Epithelial, what type of tissue blood is? Epithelial, connective, muscle, nervous. It's connective. Good job. All right. Now here we have some other properties that are really important. One of them is viscosity. Viscosity is really just a fancy way of saying thickness. So the viscosity of the blood refers to the thickness of the blood. If something has more viscosity, or if it has higher viscosity, it's said to be thicker. So think about, your Sunday morning pancakes with your maple syrup. That would have a high viscosity. Something that has a lower viscosity, if I can spell, would be thinner, more watery. So when we talk about the blood is indeed more viscous than water. Well, of course it is. Water doesn't have much in it. Some hydrogen and oxygen molecules bound together. But of course, blood is going to be more viscous than water because it has more things in it. It's got a bunch of red blood cells, some white blood cells, some platelets, some plasma proteins, some dissolved gases, some antibodies, you name it, right? So of course, the blood is going to be more viscous than water. You don't need to know the specific number, it's just there for your information. However, there could be an instance when the blood gets too viscous or too thick. Think about someone who is dehydrated. That means that you don't have as much of the plasma or water. So if we don't have as much of the water, but the same amount of formed elements, it's going to be thicker. What if someone has, what if someone has anemia? Anemia means that they don't have as many or as much as they should of the red blood cells, so their blood would become thinner. Sometimes in people that have had heart issues, we purposely give them a blood thinner because their heart is not strong enough to pump out a more viscous blood. Also, they're more prone to blood clots, so we want to have a thinner blood to prevent blood clots. Okay, so in review, we're just saying make sure you can define viscosity. It's the thickness of a fluid. And then make sure that you know that blood is more viscous than water. And of course, I'll throw in there, even though it's more viscous than water, even though it's more viscous than water, we still can have sometimes blood that's has too much viscosity or too little. All right, the second thing I want to talk about is something called osmolarity. You might hear it referred to as osmolality. And if you look at it in the textbook, in any textbook, it can often be defined in a variety of ways. And all of those ways are factually correct, but sometimes I think that they make it more complicated than it needs to be. So for us, we just need to have a basic understanding. And the way that I think it's easiest to define osmolarity would be, osmolarity is the concentration of solutes in a solution. I sure hope this is recording. Otherwise, I'll be doing this again. But that's okay. Okay, the concentration of solutes in a solution. Of course, we have to review A and P1 because we talked about solutes and solvent. These are the things together that make up a solution. The solvent is what's present in higher amounts. Everybody, say it out loud if you want. What is the universal solvent in the body? You guessed it, water. And then the solute is whatever is present in lower amounts. Okay? So osmolarity refers to the concentration of the solute. So when we're talking about the osmolarity of a solution, we're really focusing in on the solute. Let's give an example. let's say that we have saline. Saline is an example of a solution. The components of saline would be the solvent, which is water. But if we're talking about the osmolarity of saline, we're going to be looking at the solute. What is the solute? Well, what's present in saline? Water and salt. So salt is our solute, right? Because if we look at the definition here, the solute is what's present in smaller amounts. So indeed, in saline, the saline solution in your contact solution or the saline solution in a bag for an IV is a 0.9% sodium chloride and 99.1% water. So of course, the solute is what's present in lower amounts. So if I wanted to think about this, and I'm going to change color. I'm going to give a different example because I think it'll make it clearer for us. So I gave you an example of saline. We could say that saline has a low osmolarity, right? So in this example of saline, before I go on to the next example, we could say that saline, just by looking at these numbers, has a low osmolarity because there's a low concentration of the solute. Okay? Now let's give another example, one that I think will be more useful to you. Let's think about Kool-Aid. Come on now, even if you're younger, I'm sure you know what Kool-Aid is. So what are the components of Kool-Aid? Well, we have the solvent, which is water. And then the sugar powder is the solute. So in this example, we can give two different concoctions of Kool-Aid. Let's say we have Kool-Aid letter A, and then we're going to make Kool-Aid letter B. If we want a high osmolarity Kool-Aid, if I want to make a high osmolarity Kool-Aid, it simply means that I'm going to have a higher amount of sugar. So if I'm making my Kool-Aid, and let's say that the X's are sugar, a high osmolarity Kool-Aid has a lot of sugar. There's still going to be more water, the most water, but I would just simply add more sugar. And how would that high osmolarity Kool-Aid taste? It would taste very sweet. So a high osmolarity solution has a high concentration of solutes. So a good example would be Kool-Aid. If I want to make a high osmolarity Kool-Aid, it would have a high concentration of solute, which means it would have a lot of sugar. On the other hand, a low osmolarity Kool-Aid would have less sugar. How would that Kool-Aid taste? It would taste more watery. Okay, so I would encourage you to use the Kool-Aid as a good example when we're thinking about osmolarity. We're not done yet. Let's talk about the osmolarity of the blood. Do you think the blood has a high or low osmolarity in a healthy person? Well, let's draw out the components, right? And if I'm going to consider what the osmolarity is of something, or the concentration of the solute, then I need to consider what the components are. What's the solute and what's the solvent? In the bloodstream, the solvent is the plasma, which is mostly water. And then the solutes would be everything else, the formed elements. Red blood cells, white blood cells, platelets. Well, let me help you out with this. When we look at the formed elements versus the plasma, in a healthy blood, we have about 55% plasma, and we have about 45% formed elements. So here's our answer. We have 45% solutes. Is that overall a high or low osmolarity? Pretty damn high, isn't it? Excuse my language. So the healthy blood has a high osmolarity. I got 45% of my blood cells, which are surrounded by water. So it's normal. Our blood is built to have a pretty high osmolarity. Compare that to the Kool-Aid. What do you think 45% sugar would taste like? I know for me it would be undrinkable. So we like the fact that we have a pretty high osmolarity blood. And what that means is we're able to transport a lot of stuff, and it means that we're also able to bring in a lot of stuff. You can just forget this for now. Those little boxes in your actual PowerPoint are arrows, either up or down, but we're okay without them. All right, so the blood overall has a pretty high osmolarity, and we like it that way because it just means that we can transport and deliver a lot of stuff. Now, that doesn't mean that it's always okay, because certainly there can be instances where our blood can become too high in osmolarity, or our blood can become too low in osmolarity. Let's think of some examples. All right, let's think of an example where we have dehydration. Do you think somebody that is dehydrated will have abnormally high or low osmolarity? If it was me working through this, I would probably think of the normal blood, and I'd remember that we have 55% solvent, which is the plasma, and 45% cells, solutes. Let me use the same terminology. Well, what's happening in dehydration? We have our bloodstream, we got some cells, we have some water, but less water than usual. So in dehydration, the amount of cells stays the same, but I have the solvent decreasing. So we don't have as much water, but I have the same number of cells. So the overall osmolarity goes up. And this can be a little tricky. Think about the Kool-Aid example. If I keep the same number of sugars, if I keep the same number of sugars, but I take away water, is it going to taste sweet or is it going to taste watery? If I keep the same amount of sugar, but I take away water, the relative concentration of the sugar goes up. So the osmolarity goes up. So anytime you think about a time where the Kool-Aid becomes sweeter, the osmolarity has increased. So in dehydration, we have the same number of cells, but we decrease the water. So the concentration of cells goes up. The concentration of solute goes up. What's another example? I'll give you an example I gave before. I mentioned it before. Let's talk about anemia. Anemia, here's my bloodstream. I have less red blood cells than normal. Forgot the R. In anemia, I have less red blood cells than normal. So I have less solute. So I decrease the solute, but the solvent stays the same. Think about the Kool-Aid. I decrease the amount of sugar, but the water stays the same. So this is going to reduce the osmolarity of the blood. If it were Kool-Aid, if I take away some of the sugar packets, it's going to taste more watery. Okay, I know we spent quite a lot on this slide, but it's important because we're going to be talking about osmolarity throughout. Be able to, under certain circumstances, tell me whether osmolarity goes up or down. And I gave you 2 examples there to work through. This is a reminder from A&P1, won't be on the test, but along with osmolarity in A&P1, you talked about tonicity, how a blood cell may change shape, get smaller, get bigger, stay the same. If you're playing along and you had your Wheaties this morning, you already know normal saline is isotonic to human blood cells. All right, let's jump right in and talk about the components of blood. And we've already talked about this. We've already introduced it, I should say. So we know that the blood as a whole is made up of 55% plasma, and yes, I'd like you to know these percentages, and then 45% formed elements. And the formed elements are red blood cells, white blood cells, and platelets. If we let blood sit out for a period of time, or we put it in a centrifuge, it will separate. The plasma will rise to the top, and then the formed elements will go to the bottom. Also, be clear that erythrocyte is the fancy term for red blood cells, and leukocyte is the fancy word for white blood cells. Know both of them because I will use them interchangeably. Let's talk about the plasma. The plasma is the watery portion of the blood, and its big function is to act as a transport medium. 55% of the blood is water, pretty much, because it acts as a fluid medium. How else are we going to move things? Do red blood cells have little tails like sperm? Do they have little legs like centipedes that they can just walk through the blood vessels? No. So we have to have that fluid medium. So when we contract the heart, it's going to push blood through the arteries. And because these blood cells are immersed in fluid, that's how we move things. The plasma has some other functions, which we're going to talk about, but mostly let's just talk about transport medium. We are going to talk additionally about what it's made up of, so hold on to that. But I want to ask you, and I've been teaching at Hudson Valley now, this is my 12th or 13th year, I can't remember. So, I'm aging myself more and more as time goes along. It's the green slime from a Nickelodeon TV show here. What Nickelodeon show? Because when I think of plasma, I just think of like the Ghostbusters plasma. Where I think about this, the green slime from any similarly aged students out there? Hello. Yes, it was a Nickelodeon old school show called You Can't Do That on Television. All right. Don't worry about this screen. And again, when I say don't worry about it, I mean it. Okay, so this will not be on the test. All right, plasma. Plasma itself is mostly water. I'm not as concerned with the percentage here, but suffice to say, if you know that it's more than 90% water, that'll be good. So, my friends, plasma is nearly over 90% water. That small percentage that's not water is important. And these are called plasma proteins. I mean, there's proteins all over our body, but what helps to identify these are that they're plasma proteins. Good. Plasma proteins, although not numerous in terms of concentration, they are really important. Make sure you can identify the 3 primary plasma proteins, and we're going to just give one function for each. The most abundant I'm not going to ask you these percentages, but I would like you to know that the one plasma protein we have the most of is albumin. So it's the most abundant. Albumin does a lot of things. Let's just say two functions of albumin relate to fluid balance and transportation. So some things that can't be dissolved in blood, because there are things that are lipid soluble but not water soluble, some things that cannot be transported in blood will catch a ride on plasma protein, albumin. And also when we talk about fluid balance later this semester, we will talk again about albumin. All right, cool. So plasma is over 90% water for that fluid medium function. Excellent. But we also have some plasma proteins. And the most abundant, the one we have the most of, is called albumin. And it functions to help with fluid balance and to transport some things. More specifically, to transport some things that are not water soluble. We also have things called globulins, which relate to the function of antibodies. And then, of course, we have fibrinogen, which we're going to talk about later this chapter, help with blood clotting. You can imagine on our first exam, some multiple choice, I just ask you a straight recall definition of the function of each, okay? You can meet some information about at the bottom here. I'm not going to ask you that on an exam, but it's interesting. Plasma proteins are formed in the liver, and they're pretty big, which means that they stay in the blood vessels. What we're going to find is red blood cells are smaller, and they're flexible and bendy. So red blood cells are able to squeeze through the blood vessel wall out into a tissue, which is actually what we want. But these plasma proteins are larger, and that means that they stay in the blood vessels. All right, there are some other things in plasma. You do not need to know these for our exam. We do have some electrolytes, so we're going to have some sodium, particularly sodium, but I'm not going to ask you that on this test in this way. Okay, so here's what I'm thinking. What we'll do is we're going to continue to go through, and we'll talk about the erythrocytes or red blood cells, and then we'll end this video, and then we'll pick up the second half of this chapter in a second video. I tend to do it in chunks because I think, well, I'm sure you're getting tired of my voice, but it's always good to take a little rest in between so you can watch them at your own leisure, as the Brits say. Okay, so we talked about the plasma, 55% of our blood. Its primary function is fluid medium. Well, we also have formed elements, which together make up 45% of our bloodstream. And of those formed elements, red blood cells, white blood cells, and platelets, what we have the most of are red blood cells. So you should know that. Look, 99.9% of all of our formed elements are red blood cells. We just have a bunch, a ton, a million. You pick the word. So of the formed elements, red blood cells, white blood cells, and platelets, red blood cells make up the most of that, over 99%. And we know the main function of red blood cells, don't we? Transports gases. I think people sometimes just say, oh, it transports oxygen. And it does transport majorly oxygen. But as we're going to find, red blood cells also transport a bit of carbon dioxide. So transports gases, oxygen, and a little bit of carbon dioxide. Okay, we're going to talk about this more in the lab, in our first lab. And I'm sorry, my cool little units, the micro symbol, didn't come through when I uploaded this. That's okay. All right. We'll talk about this more, but just right now at a glance, when we look at the blood cell count, which includes red blood cells, white blood cells, and platelets. What do we have the most of? Red blood cells. 4 to 6 million per cubic liter. A lot. Okay, I'm also going to come back to this. Okay, we're going to come back to that. All right, so if we look at the structure of a red blood cell, it is disc-shaped, kind of like a donut. So let me get a black marker. So we can see that its outer rim is wider, but then the inner part is concave, and then it gets wider again towards the edge. That concave surface just gives more surface area. Anytime that we contort, instead of having a straight line, think of the cerebral cortex. It went up and down, up and down. Anytime we have anything other than a straight line, we increase the surface area. And that means that more function can be done. So this is absolutely on purpose. That disc shape where it's concave in the middle gives more surface area, so these red blood cells can transport more oxygen, mostly. It has a thick rim. You may look at this and say, well, it's not really that thick, but compared to other structures of similar size, it's pretty thick. It's durable and flexible. So think about, I don't know, for me, I think about a racquetball covering. You know, like a racquetball is pretty light, but if you pick it up, you can squeeze it. It's rubbery. That's what I imagine the outer part of a red blood cell to feel like. So it's durable, kind of thick, but also flexible. Something else to point out is that human red blood cells do not have a nucleus. And we see some pictures here. We're going to see some microscope blood smear images. Here we're seeing a bunch of red blood cells. In fact, the inner part of some of them is clear because this is so concave, right? So the inner part is so concave that when we stain it, we can almost see through the middle of it. Here we can see red blood cells about to squeeze out of the capillary, and they line up, kind of get into a line, and they're flexible. All right, what are the functions? Well, mostly it's transporting oxygen. We transport oxygen from the lungs into the tissue. CO2 gets transported from the tissues back into the lungs. We transport mostly oxygen, some carbon dioxide, and we also can utilize our red blood cells to help with maintaining pH. We're going to talk about that later this semester, so don't worry about that for now. All right. Now, as many of you know, when we look at an erythrocyte or a red blood cell, that looks like more of a dog bone or a humerus than it does a red blood cell. I cannot draw. That's okay. Oxygen is not able to bind directly to the red blood cell surface. So instead, we have little hemoglobin molecules in and on the surface of red blood cells. So it can't attach directly. So instead, the oxygen binds to hemoglobin. So the function of hemoglobin is this is what oxygen binds to. Oxygen cannot bind directly to a red blood cell. However, oxygen can bind to hemoglobin. So the function of hemoglobin, this is what oxygen binds to on the red blood cell. If we look up close at the hemoglobin molecule, it's very complex. It has these alpha and beta chains. That's what these are supposed to be. This is supposed to be like an alpha. This is supposed to be a beta. I'm not worried about those in particular. What I would like you to know, of course, is the function. Hemoglobin is what hemoglobin is what oxygen binds to on red blood cells. you should know that structurally, hemoglobin is made up of these large proteins. And it's made up of 4 large proteins, which tend to be 2 alpha and 2 beta chains. But each one of these squigglies, we have the gold squigglies are the beta proteins. And then the blue squigglies are the alpha proteins. But keep it simple here. All I'm asking you to know is that hemoglobin is made up of 4 large proteins. When oxygen binds to hemoglobin, it forms a molecule called oxyhemoglobin. So what I've just put into a rectangle in black is now oxyhemoglobin. When oxygen binds to hemoglobin on the red blood cell, that combined molecule of oxygen onto hemoglobin is now called oxyhemoglobin. This is a weak bond between oxygen and hemoglobin. It's reversible, but of course we want it to be reversible. So we're going to bind oxygen to hemoglobin, and then that red blood cell is going to be transported through the blood vessels out to a cell, and then we want to unload the oxygen. In other words, we want to take the oxygen off the hemoglobin so it can be, delivered to the tissues. So it's a good thing that it's reversible. Again, if you're playing along at home, this is not going to be on the test. It's just for fun, because who doesn't think A&P is fun, huh? Hemoglobin are the quaternary structures. Okay, I am not going to ask you these numbers. However, I believe it's good to do the math to bring a very important point home. Now, there's one math that I would like you to know, and I guarantee that you can handle this. Each hemoglobin, and by the way, HB is just a shorthand way of saying hemoglobin, can bind how many oxygens? 4. Know that number. Each hemoglobin can bind 4 oxygens. Why is that? Well, because there's 4 large proteins. So oxygen can bind, we can bind 1 there, 1 there, 1 there, and 1 there. So each hemoglobin can hold a maximum of 4 oxygens. Perfect. Now, if we do the math, if we say, for example, that each hemoglobin has about each red blood cell has about 280 million hemoglobin molecules. If we take 4 times 280 million, we get over 1 billion. My point is, each red blood cell can transport over 1 billion oxygen molecules. It is a lot, my friends. All right, I already mentioned that the red blood cell, and more particularly hemoglobin, is going to transport mostly oxygen, okay? But we can transport a little bit of carbon dioxide. So about 98.5% of what binds to hemoglobin is oxygen, but a smaller amount, 1.5% is carbon dioxide. And by the way, sometimes even glucose. Someone who has diabetes, who just has an excessive amount of glucose in their blood that can't be absorbed, has nowhere else to go. Sometimes glucose will kick off an oxygen and bind to hemoglobin, and that's called glycated hemoglobin. Maybe you've heard of the A1C test. That's to test how much glycated hemoglobin there is. What percentage of the overall hemoglobin is bound by glucose? All right. For each of the formed elements, we're going to talk briefly about its life cycle. You don't need to know everything on this slide, which is probably going to make you happy. But I would like you to have an idea. So red blood cells live for about 120 days, which is the same thing as about 3 to 4 months, not weeks, months. What we're going to find is the life cycle of cells is really all over the map, right? The life cycle of an epithelial cell that lines the stomach, because it's so acidic, those epithelial cells that line the stomach will only live for 2 or 3 days. On the other end of the spectrum are nerve cells, especially the nerve cells of your central nervous system. They live your entire lifetime. Well, when it comes to red blood cells, they got a pretty short lifespan. We produce them rapidly as they die off to replenish them. But each red blood cell will live on average 120 days. You can read the rest here. I'm not going to ask that on an exam. Here's a nice schematic about how red blood cells are formed, how they're broken down. We are going to talk about red blood cell formation, but you don't really need to know so much about the rest. Okay. These are just interesting numbers. I'm not going to ask you these numbers, but being that we have so many red blood cells and their lifespan is only 3 or 4 months, we need to produce many red blood cells daily. So on average, maybe, look at this, 2.5 million red blood cells are produced per second. About 1% of our entire red blood cell population is replaced each day. That's pretty fantastic. All right. I would like you to know the term erythropoiesis. Whenever we see the suffix poiesis, that means production or formation. And then the prefix erythro means red blood cell. So erythropoiesis is the process of red blood cell formation. Erythropoiesis, the process of red blood cell formation. Where does this happen in adults? You already know, my friends, in the red bone marrow. And we already know that red bone marrow lines the ends of long bones. It's in the spongy bone, mixed in between the spongy bone and the ends or the epiphysis of long bones. And in adults, we only really have 2 bones where this is still happening. So erythropoiesis is happening in the heads of our femur and humerus. And we may recall this from our A&P1 discussion of red bone marrow. As adults, we don't need as many operating red blood cell production centers because we're already grown. A child is going to be producing red blood cells in spongy bone of other bones too. But as adults, really, all we need in order to produce these mass amounts of red blood cells would be the spongy bone, the red bone marrow between the spongy bone of our heads of humeruses and heads of femurs. You don't need to know the process. Sometimes just with all that we have to cover, I can't include it all. So, okay. In terms of maintaining the appropriate amount of red blood cells, much like everything else in our body, everything operates on a negative feedback loop. And you know this, people, because we talked about it in A&P1. There are 2 ways in which our body can maintain homeostasis. The most common is a negative feedback loop, where if the level of something is altered, the body responds by reversing what just happened. So an example would be body temperature regulation. If our core temperature becomes too high, in negative feedback, we do things to bring down lower the temperature. If something becomes too high, in negative feedback, we lower it. If something becomes too low, in negative feedback, we increase it. And most of our body systems operate through negative feedback. The other option is positive feedback, which means that when something is too high, the body responds by making it even higher. So whatever change there is in positive feedback, the body responds by continuing that change. Most often, we don't want a positive feedback response. The two examples that are most common would be uterine contractions during childbirth and blood clotting, which we will talk about. But like most everything else in our body, our ability to maintain the appropriate amount of red blood cells is a negative feedback loop. So let's say that we have an abnormal drop in red blood cells. Maybe someone just had an incident where they bled. Maybe an adult woman is going through her period or menses and losing blood. Maybe you just gave blood, right? You donated blood. Anytime we have a drop in red blood cells, the blood is going to be carrying less oxygen. If there's less red blood cells, there's less oxygen. And the kidneys detect that, among other places in the body, because we also detect oxygen levels elsewhere, like in our lungs or in our carotid arteries. But one example, when the blood is being filtered in the kidney, they will note the presence of oxygen. So in this situation, the kidneys will detect, wait a minute, there's not enough oxygen. In a negative feedback loop, the body's going to try and increase red blood cell levels and oxygen levels back to normal. In this case, the kidneys that detected the change will release a hormone called erythropoietin, EPO. That is the hormone that will travel to the red bone marrow in the ends of long bones and will increase our red blood cell production. You want to see a picture? Here we go. We have a case where there's a drop in oxygen and red blood cell levels. The kidneys, among other organs, will sense that, wait a minute, there's not enough oxygen. So, in a negative feedback loop, when something is too low, we're going to do something to bring it back up. If we have a drop in oxygen, the kidneys will release the hormone erythropoietin, which goes to the red bone marrow, triggers red blood cell production, fancy word is erythropoiesis, and now we have increased red blood cell count, more oxygen, it's back to normal. Have an idea about the fact that red blood cell counts are regulated by negative feedback. I would also like to add the hormone erythropoiesis. So the hormone is what goes to red bone marrow to trigger erythropoiesis. Like pretty much everything else in our body. See if I can spell erythropoiesis. Did I get that right? Probably not. Close. Maybe I got it. Like most everything else in our body, we need these chemical messengers. So in this case, it's a hormone called erythropoietin. This is the hormone released from the kidneys that goes to the red bone marrow and tells it to increase red blood cell production. Good stuff. EPO is also very useful in other ways. A lot of cancer patients, because they're chemotherapy drugs, one common side effect is that it reduces red blood cell production. So in many instances, someone undergoing chemo will also be given EPO, either erythropoietin. Stem cell transplants, AIDS patients, but it can also be used as a method of cheating. Let me go ahead here. Yeah. Here's Floyd Landis, that's blood doping, right? So a cyclist who really relies on red blood cells to carry oxygen because they're an endurance athlete, they can actually, in addition to the EPO their body already forms, they can inject themselves with more EPO, which will go to the red bone marrow, and they'll produce more red blood cells, which can mean they can transport more oxygen and they can perform better. I also want to point out hemoglobin. One of the components of hemoglobin is iron. Okay, so in order to produce these red blood cells with what they need, hemoglobin, we need to have iron as part of the equation. All right, so as part of the red blood cell production, as part of erythropoiesis, we need to have iron. And if you don't have enough of it in your diet, you might need a supplement. More specifically, it is the iron that's needed to complete the formation of hemoglobin. I am not going to ask you about ferritin. It's just here as an FYI. When we talk about iron, once you get more into the physiology, you can't have a talk about iron without also discussing ferritin, because ferritin is what stores iron. So when you get a blood test, if someone's interested in your liver function or interested in iron, they may not only test for iron levels, but they'll test for ferritin levels. Because sometimes you can have enough iron, but you can't store it. Okay? Not going to be on the test. It's just FYI. All right, we're almost done here. At the end of each discussion of formed elements, we'll talk about disorders. Make sure that you know these terms. Polycythemia is a condition where we have too many red blood cells. Indeed, an athlete who is doping might be at risk of polycythemia. And of course, this is a problem because it increases the viscosity of the blood, and the blood becomes harder to pump. So it increases the workload on the heart. And eventually the heart will become damaged because it has to work harder just to pump out the same amount of blood. Make sure you know the definition of anemia. Instead of there being too many red blood cells in anemia, there's not enough red blood cells. And then there's sickle cell disease, sometimes associated with anemia, sometimes not. In sickle cell, instead of the red blood cell maintaining its circular shape, the red blood cells are abnormally shaped, like a sickle. And of course, when a red blood cell is abnormally shaped, it can no longer do its job, right? It can no longer transport oxygen as it could otherwise, and that's very dangerous. I talked about this before. It will not be on the test, but it's an FYI. Sometimes in addition to transporting oxygen and some carbon dioxide, glucose may indeed attach to hemoglobin. This is going to be common in a diabetic, right? There's just so much glucose. The body becomes creative with ways in which to get it out of the bloodstream. One way is that glucose can bind to hemoglobin. I mentioned glycated hemoglobin. That's what it's called when glucose binds to hemoglobin. If you do a glycated hemoglobin test, the A1C should be below 6% or 6.5, but not on the test. All right, we're going to end there, and I'll see you back for part 2.